So at the University of South Australia, we are running a Center for Drug Discovery and Drug Development, and we have basically 4 groups very strongly focused on the development of kinase antagonists, mainly for the treatment of cancer. So what we cover in those 4 groups is medicinal chemistry, we have biochemistry, we have molecular modeling, and we have pharmacology. So with this way, we can move projects forward all the way through the preclinical development phase. And this is what I'm going to talk about, give you a bit of insight what our strategy is, what we think, what we could do in order to work within the kinase field to target cancer. And this gives you a quick overview here. Do we have okay. So this is kinase talks usually start with the genome somewhere in the human genome. We have more than 500 kinases encoded, and this is where we work around. We are not doing basic research. Our projects usually start by looking out into the literature, try to figure out what other people have been doing, and a lot of information comes from knockout animals, people who generate knockout mice, and they then show in basic research that this can prevent cancer, or it can delay in animal cancer models the onset of cancer. We distinguish between hard targets. Some, like polo-like kinase is, for instance, one of those targets. If knocked out, it is deadly for the animal, it is lethal. So you can only do conditional knockouts, and we like to talk about hard targets in this case. And you can expect some adverse effects occurring, because if it is lethal to an animal, it can also be it can have some strong side effects also in human to be expected. The targets we really like to work on, we would call them soft targets. So in this case, the knockout in an animal only leads to a very mild phenotype. Sometimes you even cannot really observe a big phenotype. But you can clearly show, like with the MNKs or with the MAP kinase interacting kinases, what I will be talking about today, you can clearly show that there is a delay of cancer onset in different animal models. So our strategy, and I think the stage has been pretty well set yesterday, is to prevent a remission of cancer, to prevent resistance. We would like to target different pathways, and this is shown here. We have some projects which address the cell cycle. We have cyclin-dependent kinases as our targets, which are important at the R point, or mitosis for the cell cycle. We have one project which targets the transcription. It's also a cyclin-dependent kinase project, and what I will talk about is we think we have a way to target translation in cancer cells. So if you take all of this together, somewhere down in a few years from now, it's still quite a way to go. We would hope that we can combine the targeting of different pathways in order to prevent remission of cancer, or to prevent resistance. I quite liked in yesterday's talk that little wheel where we go around, we use different drugs, and we try to get rid of resistant cancer cells systematically. So if you look at the MNKs, where they sit, they're right down here. So this is the pathways you very often see. If we talk about cancer, you have growth factors up here. This could be EGF or EGF receptor, and you induce the signaling downstream here. This would be the mTOR pathway going through ACT, and on this side you would have the MAP kinase pathway. And those two pathways, apart from doing many other things, they seem to convert together onto a particular type of translation, which is very relevant for the translation of oncogenes and survival genes. So we already heard yesterday, we have P10 up here, which is a phosphatase which counteracts the activity of PI3K. And a lot of cancer cells, they have mutations in P10, and once P10 is knocked out, this favors tumorigenesis. So if we knock out this P10 up there in cancer cells, this leads to increased PI3K activity. In a second step, through a mutation of an oncogene, maybe RAS. This is just an example. We also activate these pathways synergistically over here. And if we have both of this going on, then we suddenly might see cancer cells occurring. What we think we can do here, and I will show you in a second why we believe so, if we block the MNKs, which are two serine threonine kinases, and the MNKs, they phosphorylate the eukaryotic initiation factor 4E. I will refer to 4E during my talk, which is the cap-binding protein. And if we prevent this phosphorylation, we modulate the translation, and we will reduce translation of different oncogenes or survival genes. And, of course, our aim would be to knock out cancer in this way. If you look at that here, this would be a helicase, and we recruit ribosomal proteins into this complex, which then starts the translation from the ATG codon. The phosphorylation of 4E is driven through the MAP kinase, through ERK P38, which directly phosphorylate our targets, the MNKs, and this leads then to the phosphorylation of 4E. And I have to mention again, this is not a black-white, it's not a turn-on, turn-off thing which happens here. It's not such that if it's not phosphorylated, we have no translation, and if we have phosphorylation, we have translation. It is much more, there's always translation going on. The phosphorylation modulates the translation, and it modulates it towards the translation of that group of genes and other genes as shown over here. We have, for instance, chemokines, we have matrix metalloproteases which play a role in cell migration. We have anti-apoptotic genes which are strongly translated after phosphorylation of 4E, like MCL, BCL2. There's the oncogene MYC, there's some growth factors, including angiogenesis driving factors, and there's quite a bundle of cytokines which play a role here, inflammatory cytokines. And I believe in the last 10, even 20 years, there has been a very strong relation has been shown between inflammation and cancer. And we believe that this can be very important in actually driving cancer. And if we block MNK phosphorylation, we might downregulate the translation of these genes, and we might at least slow down or even stop cancer from occurring. So this is the same thing, slightly different view, we believe, or it has been shown in the literature. If this here is the grade of phosphorylation of 4E, if this is no phosphorylation to very strong phosphorylation, we have at low or no phosphorylation, we have strong mRNAs like housekeeping genes, all the genes which you need in order to keep the cell alive. They are translated. If you get higher phosphorylation, you suddenly have genes coming up which are translated, driving tumorigenesis and metastasis. Okay, so our rationale from literature, it has been shown EIF4E is frequently overexpressed in cancer. It is associated with cellular transformation, tumorigenesis, and metastatic progression. This has been shown with a lot of cell-based cancer models, but also in animal models. For instance, someone made a double knockout mouse with MNK1 and 2, and it has been shown that 4E phosphorylation is completely abolished. So this, at least in mice, tells us that the only thing which can phosphorylate 4E is actually the 2 MNKs. Cells taken from these mice, they were resistant to transformation by overexpression of the RAS oncogene, and tumor formation is significantly delayed if those mice are crossed with P10 knockout mice. It has been shown in a lymphoma model that it counteracts the cancer effect. So P10 knockout mice, if this is specifically knocked out in T cells in this case, you get more T cell lymphomas, And if you cross those P10 knockout mice, this increased incidence of T cell cancer is reverted. A similar thing has been done here. someone made a knock-in mouse where they abolished the serine, which is specifically phosphorylated by the MNKs on 4E. And in this knockout mouse, very similar things have been seen. They used the P10 knockout model as up here, but in this case, the knockout has been specifically done in prostate cancer cells, and it has been shown that if this model is crossed with the S209A mutation, so this 4E version in that knock-in mouse cannot be phosphorylated anymore, and also in this case, this reverts the incidence of prostate cancer in the mouse model. So we have two animal-based models, a lymphoma model and a prostate cancer model. Very clearly has been shown that actually if we get rid of 4E phosphorylation either directly by modulating 4E or by knocking out the MNKs, this leads to decreased cancer incidence in these cancer models. And the really good thing is that the double knockout and the knock-in mouse, where we do not have 4E phosphorylation, those animals, they grow normally. There is no obvious phenotype seen, and this strongly indicates that MNK1 and MNK2 inhibitors may be effective and non-toxic anti-cancer targets. So this is basically where we started off, and this is we're not doing basic research. We get all of this, we collect out there from the literature, and we try to identify kinase targets, and from there on, that's where we start our projects. This is just for your interest, if you would like to look up those references. This is then what we do in-house. There has been the structure of MNKs has been published, and we use that for a modeling approach. We use different software tools in order to define the active site and the proximity of the active site in our kinases. We do some virtual docking. We use software for this, but we also, as input here, we also use kinase antagonists, which we already have from other targets. And very often we actually find activities on several targets, and then we try to drive our medicinal chemistry program in order to make more specific antagonists, because in the end we really would like to go as specifically as possible, which you might know is kinases. It's not always that easy. Kinases are very druggable, but usually or most of the antagonists, they sit directly on the ATP binding site, and every kinase has an ATP binding site, and this can be a problem to get really good specificity. Nevertheless, what we do then is we run different kinase assays. This is an IMAP. This is just an enzymatic assay. I will get back what that IMAP assay is. This is our primary assay to test our physically prepared compound library for hits. Then we do a secondary assay. We retest all the hit compounds with the ADP glow assay, and then we hope to find antagonists. Some antagonists might be dual active on MNK1 and 2, since the identity between the proteins is almost 80%. We would expect to get some dual antagonists, but we would also hope to get MNK1 specific antagonists and MNK2 specific agonists, which would allow us to address the question which one is more important to downregulate cancer. We then also run competition assays. We would like to figure out with ATP whether our compounds compete for the binding to the kinase with ATP. And very often we use an EIF4E-derived peptide as a substrate, and we also would like to figure out whether we have competition with the substrate. So this is just the whole combinatoric which we can get. There could be compounds with no competition effects at all. These would be allosteric antagonists. Then there are the ones which compete with ATP, the ones which compete with ATP and the peptide substrate or the peptide substrate alone. In the past, there have been a lot of antagonists described which are competitive, and we would hope, in order to get around the specificity problem, to also find purely allosteric antagonists. And then downstream from here, once we have those antagonists characterized, we routinely would run cytotoxicity assays. We would test on various cancer cell lines whether our antagonists kill those cells or downregulate the viability of those cells. We look into migration, which is, of course, connected to metastasis. A cancer cell can only make metastasis if it is capable to migrate. We test this. Sometimes there can be that these pathways which we inhibit interfere with migration. And then, of course, we are very keen to figure out the MNK structure. We have a collaboration in this direction, and we would like to know for further development, we would like to know what exactly the structure of our compound within the protein, what it looks like. This here shows our assay. So this is the IMAP assay. What we use is a peptide. This has been derived from the EIF4EC terminus. This would be the serine 209, which is phosphorylated by MNK. We have it covalently bound to a fluorophore, to a rhodamine-type fluorophore. And once this protein, this peptide is phosphorylated, it can bind, as shown here, to this IMAP reagent, which is a nanobead with a trivalent metal complex here, and the phosphate can specifically bind to this nanobead over here. And on this end, we have this linked to a fluorescence donor, which can provide, or which can transfer via FRET, can transfer the fluorescence onto the acceptor. And this means if we have high kinase activity, we have a lot of phosphorylation, we get a strong FRET signal. If we block the activity here, then we won't have phosphorylation and we have a very small FRET signal. This here just shows a series of compounds which we tested. This is MNK1, MNK2, and we have here the percent of leftover rest activity with 1 micromolar, 10 micromolar testing. You get a distribution as shown here, and this was our cutoff for hits, for primary hits, and with MNK2, the cutoff for primary hits. And what you see very early on is we seem to get many more primary hits with MNK2 than MNK1. And this sort of seems to indicate there must be some structural difference between the two, and it might actually be possible that we find subtype-specific inhibitors. We then, for follow-up, we use an ATP depletion assay, the ADP glow assay. In this case, ATP is turned over to ADP, as shown here with those triangles. We then destroy the ATP, and the leftover ADP is with an enzymatic reaction turned into ATP, then coupled to luciferase, and we generate light, and in the end, we measure light. So if you have a lot of kinase activity, we will have a lot of light down here. That's all you have to know for the moment. And with this assay, it's a totally different mechanism than the IMAP assay, and we do this sequentially in order to get rid of false positives. Because if you run biochemical assays, you will always end up with some false positives. If you use two totally different mechanisms to measure, then you are quite sure what's coming out at the end will not be false positives anymore. This is a classic enzyme kinetic. You can test with increasing amount of the peptide substrate. You can see how active your kinase is. And here again, to our surprise, MNK2 shows quite a different behavior than MNK1, which has much more activity as you keep increasing, but MNK2 seems to have a smaller Km value. And the same or even stronger if we look at ATP. If we have increasing amount of ATP, we get a stronger enzymatic reaction, and the same for MNK2, but there is a drastic difference here between MNK1 and MNK2, and this, of course, once more indicates that there might be some strong structural differences. We then use this assay to run dose response experiments. We determine IC50 for different compounds. These here are just our compound numbers down here, and in blue you see the activity on MNK2, and in red you see the activity on MNK1, and at this end we have sort of dual antagonists which are similarly active on MNK1 and 2, and then as you move over to the right here, you suddenly see this one is very active on MNK2, but much less active on MNK1, and this is probably the most extreme here. We have nanomolar activity on MNK2, and on MNK1 it is more than 10 micromolar IC50. So we clearly have MNK2 subtype specifics. Surprisingly, we never found MNK1 subtype specifics, only MNK2, and we found a whole lot of dual antagonists. So we have 7 dual inhibitors and 13 MNK2 specifics. We just arbitrarily, we decided if the activity is more than 10 times higher on MNK2, we call it a specific antagonist. If you look at the published MNK1 and MNK2 structures, you have here the ATP binding cleft. That's the active site somewhere here. And you see very clearly that there's an activation loop, which you can actually not see in the crystal structure because it's too dynamic. So dynamic parts of a protein, very often you cannot see in a crystal structure. But the end, the C-terminal end, this phenylalanine here, is the C-terminal end of the activation loop. And you can see very clearly, if you look at in MNK2, that's the corresponding residue. And you can very clearly see that the structure is completely different. So this C-terminal end of the activation loop lies completely different in the MNK2 than in the MNK1 structure. So if this corresponds to the reality, it could very strongly indicate why we see actually very different behavior of inhibitors, whether we use them on MNK2 or on MNK1. And this further encourages us to work. Also, we still would like to find MNK1 specific antagonists, which we have not found yet. So then, working towards allosteric antagonists, this is what really gets the medicinal chemist going, because there's not too many truly allosteric antagonists on kinases out there, and this is what we would like to work towards. This shows one structure. This is not an allosteric one. If you run a dose response experiment, and over here you have low concentration of ATP, and we increase the ATP concentration up to 2 millimolar. And you see very clearly at low concentration, you have a relatively low IC50 value, and this dose response curve shifts basically to the right here if you could move up to 2 millimolar ATP. So this very clearly indicates you have a competition at the ATP binding pocket, and ATP interferes with the binding of this structure as shown here. If you do the same, these are dose response curves, the same as up here, different amounts of the compound, and in this case we increase the amount of the peptide derived from EIF4E, and we cannot really see a shift in those dose response curves, and this means very clearly that we do not have a situation of competition with the peptide substrate going on in this case. Then if you just introduce a little change, this is a very similar compound. This is the compound from before. If we get rid of this part here and we add this difluoromethyl group over here, we see a quite different change, that there is even a stronger competition at the ATP binding pocket, but we also see a competition occurring very clearly with the peptide substrate. So by just changing, by having relatively minor changes of that molecule at this end, we suddenly also interfere with the peptide substrate, and this clearly tells us that this molecule somewhere sits very closely into the reactive site and can very easily, if changed, can very easily interfere with ATP and the peptide. And then this was, for us, what we really wanted to see, this compound here. We tested with increasing amount of ATP, and we could not really see very significant changes. And the same is true if we look at increasing amount of the peptide substrate. We do not really see a change of that dose response curve. And we believe that this is really an allosteric inhibitor. We had a whole series of similar compounds. This shows 2 additional compounds from that series, and we have this compound 15 here. As you can see, you have a very moderate change. If you increase the ATP concentration to 2 millimolar, there seems to be a bit of a shift going on there. Whilst if we add that group here, that functional group on the side here, we do not have that change anymore. We do not have that interference with ATP. We sort of tried to come up with a cartoon or with a working model. If this is our kinase here, you have the ATP binding pocket here, so ATP binds right in there. And if we assume that this core scaffold of that compound, which is common with both compounds, we sort of schematically show that here is that black diamond. It must somewhere bind within the reactive site. And in this case, we assume that this benzene group here can bind and interfere with the ATP binding into the binding pocket here. If we make this larger and we add polar groups here, we somehow assume that the conformation of that whole inhibitor is somehow twisted around. We still have the core binding region here, but that whole group here somehow seems to move around and it cannot interfere with ATP anymore. It can simply be steric hindering, or it can be that those polar groups somehow find other interactions with the protein around, and in this way you can get you will see true allosteric inhibition as shown here. So we had then a whole lot of different compound classes as actives, and we started to ask the question, so what happens if we put those compounds on the different cancer cell lines? And I can tell you we went into about 60 different cancer cell lines, and the only real activity we found was on leukemia cells. There were several leukemia cell lines which clearly got killed by our compounds, but most other cell lines did not get killed. And with one cell line, we systematically, this here shows the IC50 from the enzymatic assay, and we determined GI50 values. This is basically the concentration we need to kill half of the cells. So what we would predict is if our inhibitory activity is responsible for killing of the cells, we would expect some sort of correlation going on between the enzymatic IC50 value and the GI50 value. And we calculated, in this case, Pearson correlation, and we came up with a value of 0.7 and something more for MNK2 and 0.4 for MNK1, which here clearly shows that's quite a reasonable correlation. It could be bigger, but there seems to be a correlation there. Even here, where it's smaller, there still seems to be some correlation going on. And of course, if you put those compounds onto cells, we have to be aware that the physicochemical properties of the compounds, they might strongly affect passive or active transport into the cell, and this can very strongly confound actually our correlation. But if we see an 0.7 coming up here, I'm sort of tempted, it's not the final proof, but it makes me believe that probably indeed these leukemia cells we specifically kill due to our inhibitory activity of our compounds. And then, of course, you want to look into the cell. This just shows two examples. If you look at in Western blots, we expose the cells to different amounts, here shown in micromolar of our inhibitors, and then we look at the phosphorylation of the 4E protein, and you see very clearly you get a very nice dose response, and this is what you would expect to happen. Okay, if we quickly summarize what we found, we started off with 66 compounds. We had a primary and secondary screen. We came up with 13 dual antagonists, 7 MNK2 specifics. We didn't find any MNK1 specifics. We had quite a lot of allosteric. There are 8 allosteric compounds which we found. There were quite a few, but less were ATP competitive compounds. Excuse me. And if we take those compounds forward on our leukemia cell lines, to our surprise, basically all of the, or most of the allosteric inhibiting compounds, they also had activity on MV411 cells. over here there's another one, there's an MNK2 specific, and only very few, actually only one ATP competitive compound showed the toxicity against the MB411 cells. So this very strongly tells us that we're probably on the right track to go for allosteric antagonists, and we will certainly drive ahead in this way. Then in prostate cancer cells, we could not see any killing, so we chose two different prostate cancer cells as shown here. This one is a P10, they're both P10 negative. We have, according to this, ACT phosphorylation very strongly. This one is androgen dependent, this is androgen independent, and this one is not strongly metastatic, and this is very strongly metastatic. If you look at in this table, this just shows three different antagonists, and when we run GI50 experiments on the prostate cancer cells, we basically do not see much activity. We have here maybe just a little bit beneath 10 micromolar, and we have with this compound, we have very strong activity on MV411 cells. So we asked the question, why could this be? And we looked into those cell lines. Maybe I go a bit faster forward here, and we started to actually look at whether we can stimulate those cells to get a stronger 4E phosphorylation with Western blotting, as shown here, and with EGF we see a very clearly that band coming up much stronger. To our surprise, in the metastatic cell line, we do not see this at all to happen. And this was rather surprising to us. If I would have had to predict this, I would have said probably the other way around is more likely to go. But we also tested different stimulations. We looked at toll-like receptor 4, and here we see very clearly it's the other way around. In this case, we actually couldn't even pick up a band in this particular experiment with the non-metastatic cell line. And with the metastatic cell line, if we trigger toll-like receptor 4, we get much more 4E phosphorylation. So the next obvious question is, what will our antagonists do? We use them at 5 micromolar, which is clearly beneath the GI50 value. Should even not be toxic. It's totally wiped away. There's no 4E phosphorylation at all. And we then started to look into cell migration. These are transwell assays. You grow this cell line in a filter insert, and you put it into media, and you grow them on one side of the filter, and after 24 hours you count the cells, you stain them with cressyl violet, and you count the cells which migrate across. And you see without LPS you have only a few cells coming across. If we boost those cells with LPS, we have many more cells coming across. And this can be very clearly inhibited with our antagonists. And so this really encourages us to go towards this way and look more into cell migration and what MNKs, actually what role they might play in metastasis and migration. I think the conclusions are probably quite obvious. We have inhibitors on MNK1-2 dual, we have MNK2 selective inhibitors, we have detailed kinetic studies which reveal 3 types of binding modes. Our studies confirm different structural properties of MNK1 and MNK2 at the ATP binding pocket, because ATP competitive antagonists, some of them were MNK2 specific, but there seems to be a common allosteric binding site. MNK inhibitors are cytotoxic against some leukemia cells, but not against most other cells. No cytotoxicity has been observed against prostate cancer cell lines, but MNK inhibitors, they block the LPS-induced migration of prostate cancer cell lines. Sorry for being a bit long, but this is the end of my presentations. These are all the collaborators, students, and postdocs who did the heroic work on this, and I'm open for questions. Thank you very much.